All right, ladies and gentlemen, let's do some metabolomics. So what I'd like to do in this last part, this last presentation, is talk a bit about metabolite identification, and then you can continue to work your way through the tutorials, in particular with that in mind, what we are discussing right now. We have been talking about the size, the potential size of the metabolome. Yesterday we have been talking about the differences in structure, what that implies for quantifying metabolites. When we compare proteomics and metabolomics, then the world of metabolomics is much more diverse. We are dealing with structures, no longer with linear sequences. We are dealing with very different ways in which metabolites can fragment. We will discuss that in a minute. Where we saw that peptides basically break at the backbone, at the peptide bond, or to the left and right to it. Metabolites can basically break in very arbitrary ways, and oftentimes the spectra are not as informative. They have fewer peaks because the distribution of bond energies is different than in peptides. You end up with 5 or 7 peaks that do not convey as much information as you would like, or as you're used to from peptide spectra. The first thing you can ask is, well, if we know so many metabolites already, can't we just go through databases? And that is exactly what we do. There are different types of databases for metabolites, and I would like to introduce them to you. At least some of them. There are dozens of databases that have a certain size. You can basically break them down into 3 distinct categories, and many of these databases contain more than one category. These categories are structural databases. They contain the structures of metabolites. Their pathway database that contains also the relationship of these metabolites in metabolic networks. And there are also spectrum databases that contain information on mass spectra, experimental mass spectra, but not only experimental, some of them also contain theoretical mass spectra. In contrast to sequencing, where basically everything that is being sequenced is put out into larger archives and is freely accessible except for personal genomics data. Freely accessible spectrum databases for metabolites are rare, and many have parts of them that are still proprietary, or that you have to buy licenses for. They're being sold commercially, and that has been hampering the field of metabolomics for many years. The first database I would like to point to is HMDB, the Human Metabolome database. That is a database of known human metabolites. So if you're looking for secondary plant metabolites, that's not your place to go. But if you're working with human cell lines, that is a good resource. Because similar things hold that we discussed for proteomics identification. You should match your search space to what you're actually trying to find. Otherwise, you can be misled. And the same holds for metabolite identification. If you know that you're working with human metabolites, then you should probably not go out and search for plant metabolites unless you have a very good reason to do that. It's a very well done database, nice web interface. You can go in there, you can search for metabolites, for example, for glucose. That's what I did here, and then you get a couple of entries that have glucose somewhere in. Of course, the top hit is actual glucose. You get the molecular weight or the monoisotopic mass as well, the formula, and then you can click on that and you get these so-called metabo cards. They're basically the key records in this database. You see the structure of that compound. You see that there is some annotation, some meta-information on what they call a chemical taxonomy. So the kingdom here is organic compounds, the superclass carbohydrates, class carbohydrates, monosaccharides, mammalian metabolite, and then you see there are a few annotations that refer to the chemical structure. It's a heterocyclic compound, a secondary alcohol, hemiacetal, and so on. So you get some additional information on what you're really dealing with on the chemical side. And if you go further down, you see the average molecular weight, the monoisotopic molecular weight that we discussed, and the smiles. And the smiles is kind of useful. That's a string representation of the structure. And you can, for example, copy and paste these smiles directly into KNIME. And KNIME will render the structure and can deal with these structures in that way. You can just copy it from there. The other thing that you can do with HMDB is you can go out and search. What can you search? Well, there are different ways to look at that. The first thing you can do is you can just search by accurate mass. So if you have a feature that is differential and comes out of the analysis we did yesterday afternoon, you get a list of features and say 10 of them are interesting. You just look at the mass of this feature, the monoisotopic mass that you can extract. It's part of this table that comes out of the OpenMS analysis. And you just paste that number into this search field, and this will then perform an accurate mass search. It will match your measured accurate mass against all the exact monoisotopic masses in the database, and you will get a list. And as you can see, this list is pretty long. Why is it so long? I used a mass tolerance of 0.1 Dalton. That is probably not what you would want to go for if you have a high resolution instrument, but it gives you some idea that just searching by mass is going to give you plenty of potential candidates. Now, using glucose is also a mean compound in that respect, because all the hexoses will have exactly the same molecular weight. So you get plenty of candidates, but you see glucose is here on top, fructose, of course, glucose, mannose, galactose, and so on. So everything you would expect, but also a few unexpected things. Theobromine is also here. Aspirin is in the same molecular weight. And you see this is sorted by mass deviation. So as you go down this list, it just becomes less and less likely that you have the right hit. You can also see the compositions here. You see the first hits, they are closest. They're all C6H12O6. So it figured out the right molecular mass, or the hits have the right molecular mass for glucose, which is not surprising because that's what we started with searching. You can click on these entries and then get additional information. You can look at experimental spectra as well. So there are spectra stored in here, not just MS spectra, but also NMR spectra. If you look here, you can look at the 3D structure of these guys. You can look at predicted NMR spectra. And there are also mass spectra, and that is what's interesting to us. Some of them are recorded on different instruments, on different collision energies, and you can download these spectra, but unfortunately you can download it only one by one, so you cannot sort of do a bulk download of the database. You can, however, search with the full spectrum against this database. So in the same way that in proteomics identification we use the peak list and compared it to theoretical spectra, you can also compare to experimental spectra. Since you cannot download HMDB, you have to go through the web interface all the time. And that makes it a bit clunky. It would be easier if we could integrate that into the workflow, but since we can't download it, that's the only way to actually do it. I don't know why they do it. But that's the way it is. And that is why it's essential to do the quantification first, really see what you are interested in, and then you end up not with 10,000 of spectra that you might want to identify, but then at that point you have to go through their interface if you want to identify them. That is what they call the MS-MS search. It's, again, a simple web form. You basically provide information on the parent ion mass and on the tolerance. And you say, I saw a parent ion at this range, and the more precise that is, the better it is, because it will use this mass, the accurate mass of this precursor, to basically cull down all the experimental spectra. And then you need to provide the spectrum. And the spectrum, that is a very straightforward format. You just paste a peak list. So that's M over Z intensity, M over Z intensity. Basically one peak per row. You paste that in and hit find metabolites, and off it goes and returns a hit of a spectrum. Then you can look at the spectra and see whether you're going to believe it or not. And ultimately, the proof's going to be in the experiment. You would probably see that you can acquire that compound, run it on the instrument that you ran your original experiment on, and see what the spectra look like. Because on different instruments, you might also get different spectra. The second database that I'd like to highlight is Metlin. That contains a very large number of metabolites. Last time I checked, more than a 250,000. And not for all of those, they do have spectra. Some of these metabolite structures are just taken from literature. But for about 12,000, they do have spectra. And they permit search of metabolites via their mass spectra in a very similar way as you've just seen it for HMDB. So you just submit a peak list, it goes off and retrieves all the hits against that. A very convenient database to search is also MassBank. It contains lots of spectra. It's mostly older spectra, however. It has been growing significantly in recent years. There's also a nice R interface, so you can query mass banks through R. It's quite convenient to use. It just doesn't contain as many metabolites as Metlin does, for example. Then, more recently, the colleagues at UCSD started implementing a database that is called GNPS, Global Natural Product Social. And that is a place where you can upload your spectra. So the idea is you just don't keep your spectra, you upload everything that you care about, and perhaps also the rest. They actually appreciate when you upload all your spectra. And they will try to match these spectra and identify who else has seen the spectrum. And the idea is there's a community effort. You know, if someone identifies their spectrum and says, you know, after 2 years I figured out this is X, then you can add the annotation, and then all the other guys who have seen this spectrum or a similar spectrum will know this looks like it's X. And that is a very valuable effort. It's also very well done. And I can only encourage you to go to GNPS and try to upload your spectra there to search for it. Then there is Mona. That is an extended version of MassBank that's maintained at the Fien lab at UC Davis. Also has a nicer web interface, more spectra in there. Otherwise very similar to MassBank. And there are, of course, commercial databases. One of them is MZ Cloud. It's technically open. It's actually run and paid for by Thermo Fisher. It contains only a limited number of metabolites, but basically everything you could buy from Thermo is in there by now. And they recorded it very diligently on, surprise, surprise, their own instruments. So they did that in a very consistent fashion, very high quality spectra, high resolution spectra. But again, you can't download it. You have to go through their interface, have to go through their software if you want to access it from the instrument itself. But otherwise, neatly done. You might have used this one, KEG, the Kyoto Encyclopedia of Genes and Genomes, that is mostly known as a metabolic pathway database. I've actually shown one of these images. Remember on the first day I showed you the citrate cycle that was a KEG map. They have not just these pathways, but for every compound in the pathway, they of course have the structures, they have additional information on all of these compounds. They do not have spectra at this point. Then there's human psych and eco-psych. That's a whole range of databases. They have a very comprehensive set of metabolic pathways, and in contrast to KEGG, you don't have to pay for a license if you want to use the database. Yeah, KEGG used to be free. It's no longer entirely free. If you want to download it, you have to have a license as well. And finally, often overlooked, PubChem. PubChem contains basically everything we know about chemical compounds. So over 30 million compounds that are in there, and you can just go into the database and see whether you can find something that has a certain composition. You can also go in there and search by monoisotopic mass if you care. The problem really is with PubChem that since it contains everything that is known in chemistry, you might find hits that are just not what you want. They're just very weird compounds in there that just have been described once that are unlikely to be metabolites. So you got to be careful. It's just exploding your search space, and you might not want to do that. But if all the other databases fail, then go to PubChem and see whether you can't find anything that fits the bill. These databases are growing quickly. It's hard to get accurate numbers, so I apologize. I didn't update them this year. It's a tedious work, and it's going to be outdated the next day. So I just give you some idea of where they are. PubChem had 63 million compounds. MZ-Cloud had 2,000. Metlin, you see, is the largest by far, and HMDB had 41,000. And what you also see is that the numbers are growing very quickly. I'm not sure they're growing exponentially, but they're growing very quickly. So the odds that you are going to get an identification are getting better by the day. And that means it's also worthwhile that even after you did your search and you didn't come up with anything meaningful for the identification, and then 3 months later you rerun that stuff, you might get additional hits. I mean, it's not guaranteed, but it's definitely something to keep in mind. Since the stuff is growing, just rerun your identifications. In that respect, it's also essential that we increase the data that's out there. And that is also your responsibility. If you run metabolomic studies, or proteomic studies for that, please deposit the data. I cannot stress how important that is. First of all, it is essential for reproducibility. If you really want people to believe what you published, then you should get your primary data out there. And that means upload all your spectra. Please do not just add a supplementary material, an Excel spreadsheet that contains some identification that you believe to be right. Please upload all your primary data. So for metabolomics, there are a few repositories. Probably the most important one is Metabolites. Metabolites is hosted at the EBI, European Bioinformatics Institute, and it stores the original experimental data and as well as the interpreted data. So you can upload just your raw files, your MZML files, or whatever you generated to analyze the data, but it will also hold your interpretation. For example, the quantification results, the identification results. And that is where OpenMS comes in handy, because you can generate, with these workflows, you can generate MZTab files. And MZTab is the standard format that can be used for data deposition in metabolites. I know that who has ever deposited data in metabolites? Pride? Massive? Wow. All right, then I take 5 minutes and talk a bit about why you should be doing that. First of all, depositing data is a pain in the butt. because you really need to pull everything together. You need to provide metadata. What organism did it come from? What instrument was it acquired? What was the purpose of the study? Which are technical replicates? Which are biological replicates? All that. And it's going to take you a couple days to do that. So what's the benefit of depositing the data? Or why did you guys deposit the data? Because editors forced you?
No, I think it was just like our field has sort of started doing that.
Yeah. That's good. That's good that you're doing that. What are the advantages of depositing data? What do you think?
Did you have to take your study?
Yeah, you might argue, I don't want my study to be replicated. Yeah, I don't want to see other people see what I did and possibly find a mistake in there. Yeah, that's one of the things I hear quite a lot. Yeah, I don't want to give other people an advantage, and I don't want other people to sniff around in my files and see whether I got it wrong. To be honest, that is a total intellectual capitulation. Yeah, if you don't think that your publication is good enough to withstand somebody looking at it, then you shouldn't be publishing it. The second thing is, how often have you been upset by other people where they wrote data available upon request and you write email after email and you get nothing, exactly nothing. You get if you're lucky, you get a polite email, yeah, I would like to give you the data, but the PhD student moved on, died, got run over by a car, I don't know, and I can't find it because we put it somewhere and we can't find it. And it's so basically, academic labs are pretty poor at data management. And I can guarantee that because I'm running a core facility that does data management, and it's messy. People believe that if you just burn a DVD with your data and put that into a closet, then you have done due diligence. Far from it. That's actually, as far as I'm concerned, that is scientific misconduct. So how can you do that? You can either run an infrastructure that takes care of your data. And by taking care, I mean really have annotated metadata, have several copies of the data, so in case something goes wrong, you know, a water pipe bursting, and suddenly your server is 3 foot underwater. That has happened. Most labs just cannot afford the staff and the money, the hardware to actually set up this type of infrastructure. So the best way is actually to go to one of these public repositories and say, you take care of the data for me. And then you're rid of it and you've done due diligence. And all you need to provide is somebody sends an email, where is the data? You just say, Metabolites, ID, MTS 435, and go there, have a look at it, have fun with it. The other thing is, if you do that, you will get more citations. People will use your data. Bioinformaticians, I have been called a data parasite, but you know, that's the whole idea of science. You build upon what other people have done. It's not parasitism, it's actually making good use of the money that other people spend. So you can go out there, you'd publish a paper on the data that someone produced for something entirely different. Remember when we were talking about feature finding for metabolites? How did we do the synthetic data? We took a published experimental paper of people who were so kind to actually provide all their identifications. We took their data, synthesized the data set, and of course we cited them. And if you want your data not to be in vain, then just deposit the data and make it accessible to other people. End of rant. Raw data repositories. In Metabolites, you can go in and get an arbitrary Metabolomics data set, all the raw data, all the metadata through what's called an identifier. For example, here, MTBLS17. What you get is a short description of what this is about. So utilization of metabolomics to identify serum biomarkers for HCC in patients with liver cirrhosis. you got a few, basically a brief abstract that describes why that study was done, why people thought that might be a useful thing to do. There's not always a paper attached to that. People also deposit things before they have been published, or things that they did not publish for whatever reason. And you have additional information. You see the data is from human, it's from HCC, it was produced by LCMS, and for every study there's information on each of the samples and runs. And now you see why it's so painful to actually upload that, because that's the information you will also have to provide. There's actually a tool there in Metabolites that helps you to assemble this information. So it's, it's not terrible, and you should have all that information in your lab notebooks anyhow, or ideally in something smarter than paper. And that makes it easy for you to navigate this, and you have additional information, for example, that this was acquired on Acuity UPLC, that was an RP chromatography, and things like that. And then you can go in and you can just download all the data for the various formats from that instrument, and you can also browse the identified compounds that were in this data set. So it's not only useful to go and look for someone's raw data, but it's also useful if you want to know who has found a certain metabolite in liver cancer. Then that is something you can do here as well. And you can do that only because people have really deposited their data with the full metadata. There are other repositories that do not require metadata, but they are slowly starting to understand that without the metadata, that what they deposit is only half as valuable. Don't get me wrong, it's still better to have the data without the metadata than not have it at all. But it's of course much more valuable when you have some context on how this sample was produced. So you can go in and search by an organism, you can search by mass spectra, you can search by a database, and the idea would be now if everyone deposits their data there, then this collection of data is growing very fast. And if you remember the first lecture I gave, I had this super exponential growth. That was metabolites. So people are really depositing the data there, and that is boosting the value that you have in this database. Okay, yes?
Can you get a database from Metabolites? Can you download the data and use it as database?
That's a terrible question. Yes. So the short answer is yes, but of course you can download everything. The problem in a way is that it contains information from different instruments in different formats. So this is not just one relational database that you can download, but it's a huge data dump. It's all connected through this metadata, but it's actually very hard to navigate the information. But you can get it from EBI. Let's talk a bit about what these databases are doing under the hood, because so far I've only shown you where you can search for that, but what's really going on. So metabolite identification is a non-trivial topic, and it has been holding the field back for the last 10 years, and only for the last 3 or 4 years we have been seeing significant progress. And I think in a couple of years this identification in metabolomics will be standard through database search in the same way that we're doing it right now in proteomics. So what people have been doing a lot, and that has been going on for decades, is just identification by accurate mass. So if you have a sufficient mass resolution, the mass alone contains information on the metabolite. It's not going to be a unique identification. You've seen these lists that I gave you for glucose. And the problem here is that we are in a narrow mass range. We're dealing with masses up to 300, 400 Dalton. And then you have such a large number of possible structures that you run into lots of structural isomers. How can you get out of that? Well, you can always look at the tandem spectra. The tandem spectra contain more structural information than just the composition. And the composition is basically what you match with the accurate mass. So you can do that with spectrum comparison. And traditionally, when I was studying 25 years back, what we did to identify mass spectra was compare them to spectra in books. Yeah, so there was a huge index, and you just went in with the mass spectrum with the 3 highest peaks. You index that, and you check, does the spectrum look like that? And that's basically what we're doing these days, except we don't use books anymore, so we use spectrum libraries. So we compare the experimental spectrum against the theoretical other experimental spectra, and that requires libraries of experimental spectra. Many labs have built up their own libraries in-house. They just buy 500 compounds and run them through because those are the ones that they're interested. They think that's a highly valuable resource, so they don't share it. So everyone has 500. And that's, of course, very, very inefficient. And, of course, they're not as valuable as people think, because everyone has them, so it can't be that great. But since nobody's sharing the data, everyone has 500 different ones. The overlap's probably going to 400, because everyone does amino acids, everyone does a couple of sugars. But since we don't have this culture of data sharing, people search with their own small in-house library rather than searching with a large library of everything that is being out there. But still, it does work. And more recently, people have started looking into so-called fragmentary approaches. And fragmentary approaches are based on structural databases. Fragment trees or fragmentation trees try to predict the fragmentation patterns of a given structure. Now, these structures are freely available in the databases. You can download PubChem. You can download all the structures from Keck, for example. That is readily accessible. And what you can do is you can compare your experimental spectrum against a theoretical fragmentation pattern. That's basically what we did in proteomics, except that in proteomics, predicting these spectra was trivial, because it either broke at a peptide bond, you get a B ion, you get a Y ion, done. The whole thing is linear, there's not much to do. But if some of you have ever done gas phase chemistry in mass spectrometry, either theoretically or really worked on the spectra, you know that funny things are going on in mass spectrometers. There are rearrangements, things break off in unexpected ways and at unexpected places. It's very hard to predict mass spectra, to be honest. But we will go into that because this is where we had the most progress in the last 3 or 4 years, and this is probably going to be the future of metabolite identification. So let's look at accurate mass search first. There's an oddity that many people don't expect, namely that the metabolites are distributed over the mass in a very uneven way. When you look at the number of hits in a certain mass range, then it's not unexpected to see that for low mass range you have few hits, and these hits get, the number of hits gets much larger if you go into higher mass regions. Why is that? Because you have structural isomers that have the same mass, they all fall into this mass bin. What's a bit unexpected is if you zoom into one of these mass ranges, going from 100 Dalton to 101 Dalton, you see the vast majority of hits is between 100 and 100.1 or 100 and 100.2 hits. And the rest is basically empty. Why is that?
Mass defect.
Yeah. We're dealing with a discrete distribution. The masses, the monoisotopic masses of these compounds are predefined quantities that are given through the isotopic masses. So no matter how you arrange them, they will always add up to multiples of these element masses. And that gives rise to this funny mass distribution that you see. When you zoom in, I mean, this here is just the number of potential decompositions of a mass. So how many compositions can you build out of that mass? And you see that that goes up exponentially, not unexpected in the mass range. But if you zoom into a certain mass range, here around 1000, then you see it has this funny behavior, and that just comes from basically the last digit that you have in the molecular weights of this composition. So there are some masses that are way more likely than other masses, and that, of course, hampers the identification a bit here, because you end up being in one of these highly frequent masses more often than not. When we go into one of these masses, like we just pick 0.790, that's C5H11NO2, then you see that there are already plenty of hits. And some of them are structurally similar, because we know we have at least two oxygen. We have exactly two oxygens, one nitrogen. You can think about how many possible arrangements can there be, and the answer is quite a few. You can see you can construct a nitro group, but you can also construct a carboxylate with those two oxygens. Then you have to have an amine here. So there are different ways to go about this. What we see is that these metabolite masses are centered around 300 Dalton. And if you hit this range, then of course your identification with an accurate mass becomes very ambiguous. So rather than having one good hit as you would get out of a proteomics database search, here you often end up with 100 hits. It depends on the accuracy of your mass spectrometer. If you have a higher accuracy, you can sort of narrow it down to one composition. If you have lower accuracy, you end up with more than hundreds of candidates, because you might have more compositions that match your accurate mass. So a second approach was, let me finish on that. People have tried many things to increase this accurate mass search, but basically once you're at the composition, you have to deal with this ambiguity. And we discussed that yesterday a bit. You can try and go for retention time. That is what people do in gas chromatography a lot, GC retention time indices. But again, when you look at these structures, it is not implausible that structures that are very similar, like those two guys here, they have the same composition, but you would argue, well, their retention time is not going to be vastly different. They're chemically very similar. You might exclude some of these structures, but not the majority of them. So a much better way is to provide information on the structure, because the composition alone, that's all you can get from the accurate mass. If you want to know what substructures are contained in there, then you need to fragment the thing. Now, you can't control fragmentation in a meaningful way. You can, of course, modulate fragmentation, the collision energy, and get different types of spectra, but ultimately the molecule is going to break in the way it wants to break, and you have to deal with that. It's going to give you some information. It's not going to give you the full information. The same way that we do not always see all the B ions, not all the Y ions. We see only certain fragments that arise from the structure. With typical MS-MS searches, we can still make use of that, because they will fragment in a consistent way. Yes?
I think I got the raw big data from HMDB right now. Yes, I think it's available.
All of it?
Yeah.
Okay.
I think so.
Then they changed it. But check whether it's all of it. It used to be only a subset.
Like 600 megabytes of the Eastern spectrum.
It's possible, but it used to be incomplete.
I think it's good, yes.
Some things are moving forward. So what you can do is you can try to see whether the tandem spectra are similar to what other people have recorded. And this is how you do this with Metlin. You just copy and paste your peak list. And then it goes off and searches. And it returns a list of potential hits. And it also shows you the spectrum. So on the right-hand side, you see these mirrored spectra that you've probably seen. You can also do that in top view. And you can see, yeah, these peaks look exactly what I have seen, or are reasonably similar to what I have seen, that I believe this identification, or as in the second case, it's probably something different. It also gives you some idea how sparse these metabolite spectra are. There are not that many peaks. So they are not as information rich as you would like them to be. What Metlin does underneath is roughly this. And you see this is looking very similar to what we were discussing for proteomics, except that we're now matching experimental tandem spectra with experimental library spectra. So rather than taking a sequence or a structure and predicting these spectra, we just store the spectra. And now you need to be careful because one of the big issues is you need to have a similar fragmentation, a similar instrument that was recorded on. And depending on how you do that, you might get no hit or might get wrong hits. So that's something to keep in mind, and you need to be careful with your metadata how you select that. But once you have candidate list, what it does is very similar to the proteomics database search. It has a metric that is usually based on shared peaks or some sort of correlation of the spectra. There's some statistical ways to turn these into meaningful scores, and then you get score hits in the same way that you know it from PSMs. So this is basically the equivalent of a PSM in library search. It also means you will always get something back, almost always, and you need to figure out how to do that. And here Metabolomics has been tailing this whole field a little bit. Only last year we had the first one, and the year before we started having the first papers that were considering how to do FDR on this. FDR is a lot harder because we're dealing with a more complex space than we have in say, structure space, but also it's much harder to construct decent decoys. You just can't invert a structure and say this structure can never occur. So that was a real showstopper. We didn't have good decoys, and if you have decoys that are far away from what you would have, then your FDR estimates on that. So now there are first papers that try to estimate that. So in spectral library search, we are really trying to identify the closest experimental spectrum that's known. You need to have libraries that you can download, or your in-house curated libraries or commercial libraries that you buy. There is a top tool that can do that for you if you like. It depends on the instrument type, on the instrument resolution. All these things need to be carefully controlled. If you've ever tried recording tandem spectra of metabolites and varied the collision energy, you have some idea of what that looks like. Many people for this reason also record the spectra with different collision energies, and also these libraries record them with different collision energies, and then you hope that you match one collision energy that is closest to what you see or you used and get a hit out of that. Spectral library search has a few issues, and that first issue is of course that the metabolites are often less information rich, and not all the databases are readily available. But there's also another issue: not all of these compounds are available. So you can imagine if you're doing natural compounds, anyone who has worked in organic chemistry trying to synthesize natural compounds is a non-trivial task. So you cannot just go out like with peptides and buy a synthetic peptide. You need to find these compounds in order to record their spectra, and that is often a big deal. You just can't order them online. Accurate mass search works in a different way. Here we are actually not searching against experimental data, but we're searching against the metabolite database, usually a structural database. That could be something like PubChem. And we take all these structures, and from them we compute the monoisotopic masses. But we have to add something that we haven't discussed yet, and that is an adduct list. Most of these monoisotopic masses we will not observe as the masses themselves, but they will have adducts like sodium, ammonium, or acetonitrile that is being attached to this metabolite, usually as an ion pair. And what you do in accurate mass search is you take all these monoisotopic masses and you add the adduct masses, And for each of these possible hypotheses, you ask the question, is the M over Z below the tolerance that your instrument supports? And if that is the case, you get a hit list. Note that you do not have a natural score for this hit list. Yeah? So it's just everything that matches in this threshold. You can, of course, say, well, those that are closer might be more likely, but that is not exactly true in many cases. So what you get is just a set of potential candidates, and then you have to start thinking about this set and start thinking about how can I narrow it down. And then, of course, mass spectra can help, retention time information can help as well. If you do that in Matlin, for example, you have to provide some additional information. So there is, of course, the mass, there's the tolerance that we just discussed. You need to know whether you were in positive or negative mode. And then you can just select the adducts that you believe are likely. And that is something you need to know from your experimental conditions. How much salt was in the buffer and what was in the buffer. That is something you need to know. If you know that, you just check these potential adducts and you hit search, and again you get back a list. And you see here, this is the closest that we have to a score. This is a delta ppm. That's the deviation. That's the difference between exact mass and accurate mass. And you would, of course, hope that if you're very far away from the exact mass, then it's unlikely. And you would hope that your accurate mass was closest to an exact mass. Okay. There are some caveats with accurate mass search. Limiting factors are, of course, instrument accuracy. Big mass error implies huge search space, so if you don't get that exactly, then you're in trouble. And that means you need to have a good idea of how accurate you really are in one way to do that is if you have known compounds in your data set, then you can determine your errors that you see. You can also look at things like the distribution of all these metabolites that you identified through library search. You know, if you know I'm always going to find these 50 in there, you just identify them with library search. You know where they come in terms of retention time, and you can actually determine the distribution of your error of these masses on your data set itself, and that gives you a better estimate than probably what your mass spectrometrist is going to tell you what they see, what they expect their instrument to perform. Sometimes it's going to be bigger, sometimes it's going to be smaller than that. But the bigger issue is really database completeness. We still search against these databases where we don't know how large the metabolome really is, and we're still discovering new stuff every day in metabolomics. So you're restricting yourself to a subspace of the chemical space that's out there that might not be what you're looking for. Also, accurate mass has this issue with this isobaric ambiguities. There's just no way around that. Now, how would that look like in OpenMS? I've shown you this pipeline yesterday. Now you have some idea of what's going on under the hood. We have one tool that's called Metabolite Spectra Matcher that does exactly what you would expect it to do. It takes a spectral library and it compares all these spectra and gives you the best hits based on the scoring. You can filter those hits, you can look at the scores. And many of those spectra that you have in your input data set will not get an identification through that. Much of or the majority of the metabolome will be unknown. And for those, you can then use accurate mass search. What we provided there is a list of compounds taken from, for example, mass bank. You have these compositions. You search with the accurate mass search. After you define a list of potential adducts, you run that, and then here's this joiner node. And what the joiner node does is it takes these results and it merges them together in a way that you have both hits for every feature, for every original tandem spectrum. You have everything you know about the accurate mass, and then you can sort of go over that list. And if you find if you find a library hit that looks okay, and you have also an accurate mass hit that includes that compound, then that's already a good thing. But if you don't have the library hit, then you can start thinking about whether you take the spectrum, the peak list, and copy it to Metlin and search there, or go to other databases and see what you can do about it. This node here, RDKit structure, that computes the structures for the database entries, so that you can then look at this interactive table, and you've seen that yesterday, this nice scrolling view that's constructing these representations of the structures back here, so that you get, if you're a chemist at least, that's easier to read than just the name of the compound. Okay. Perhaps the most interesting part are fragmentation trees, and these fragmentation trees are a computer sciencey way of thinking about structures. Technically speaking, fragmentation trees are not really trees. As we will see in a minute, they are directed graphs relating the molecular structure of a metabolite to potential fragment masses. So the idea is fragmentation means breaking a bond in a metabolite. So if you have the metabolite structure, if you take out one bond, then there are two options. It can either fall apart at this bond, or if the bond is in a ring, you get a new structure that can sort of break apart. Breaking rings, of course, requires breaking two bonds for the structure to decompose. And by systematically and repeatedly breaking all bonds in the structure, you can sort of think of what would happen if I break this bond. Now, of course, not all bonds in the structure would be broken. All bonds are not equal. They have different bond strengths. Some bonds are more likely to break than others. And it's not easy to predict that. When you try to do that, you end up very quickly with doing quantum chemistry. And that is something that doesn't scale very well. Having started out as a quantum chemist at some point, I can attest to that. Fragmentation is a stochastic process, but it strongly prefers low energy dissociation pathways. So you try to activate the bond, and sort of the weakest link in this molecule will break. But of course, you can also activate higher ones. It's all a matter of energies, and that's where the collision energy comes in. At different collision energies, different bonds may break. The problem now is that if you do that theoretically in the systematic manner, you get lots of fragments, but the majority of those will not be observed. You saw the spectra. There are only like a handful of peaks in there oftentimes. And that has to do with the gas phase chemistry. There are rearrangements that lead to different structures. There are neutral losses that are not easy. And of course, the cycles pose a problem. How do you deal with these cycles? There are different ways to construct fragmentation trees now. One philosophy is really decomposing the molecular structures. You can say, I take this molecular graph, atoms as nodes and bonds as edges, and I just cut these edges, and whatever comes out is a new graph. But you can also think of this about molecular formulas. Since all we can measure from an accurate mass is really just a formula, you can think of ways of how you can decompose molecular formulas. I'll show that idea in a minute. The first method requires that you have information on the molecular structure, but also leverages that, so you get more information. But the second approach relies on just sufficiently accurate masses, and that is fair enough because it's not biased through structures in a way. This, you see from the dates of the publication, this is rather recent, that this whole area started about 10 years ago and then started to pick up speed. And initially people did not really know how to model these things. The accuracy was pretty poor, but it has gotten a lot better since then. So this first idea by Heino et al., they suggested to start with a molecular graph and just walk all the edges and decompose the graph. So you start with a structure, and you think about possible ways on how to break this. I could break this bond, this bond, this bond. Depending on how you do that, you end up with different fragments. Each of these fragments can then be broken down further. And you can, of course, if you pick this guy, you can break all the bonds in there and end up with a different set of fragments again. And that's the way this fragmentation tree is formed. I will not go into the computer science parts of that in too much detail, but the paper is easy to read. I can only encourage you to go into it to get an idea of what's going on. This fragmentation tree basically tries to be as unbiased as possible. At the same time, they need to put in some information on what are the bonds that are likely to break. So you can sort of attach something to these edges and say these edges have a certain probability of occurrence. Finding a good function that models this likelihood, that is actually the hard part. Because to do it properly, again, you would need to do quantum chemistry to predict that, or you can have some simple rough cut rules and say certain bond types are just easier to break. Or you can start and think about learning this information from larger databases. Again, that is where large databases come in. The more data we accumulate here, the better we get with this type of a prediction. Ultimately, these fragmentation trees predict something that is sort of the space of all possible fragmentations, but what your experimental spectrum gives you is the most likely fragmentation. And that needs to, there's a certain disjoint here that leads to problems. The second thing you can do is structural fragmentation trees, where you decompose repeatedly these structures. The results can be scored, and the idea here was to find the lightest subtree, the subtree that breaks the fewest bonds and still explains all the fragments in the experimental spectrum. And that gives you some idea of how the spectrum came about and what you should expect in this particular substructures. It turns out that finding this lightest fragmentation tree, unfortunately, is a pretty tough problem. For the computer science guys, it turns out to be a mixed integer linear program. They tested that, and for the majority of metabolite spectra, their method could explain 80 to 90% of the fragments. So that's not too bad, but it just doesn't scale very well. If you think about looking at large libraries, then this is just not fast enough. Also, they didn't really provide a tool that you could use that, so this paper remained a little bit academic at that point. Another idea are compositional fragment trees. Here you're actually not starting to think about what the structures are underlying this, but you're looking at compositions only. And in a way, this is similar to de novo identification in proteomics. You're not biasing yourself by a structure database of which we know that it is incomplete, but instead we're just looking at the composition that we see. And from the accurate mass, we get a decent composition. And then we can start thinking, how can you decompose this composition into other possible structures? And remember what we saw about the mass distribution. All masses are not created equal. Every one of these masses can only be made up from different subcompositions. So you can, that's a reasonably well-known problem, you can just take these masses apart and come up with a way to decompose them. So let's look at that. I hope you can read that from the back. You start with the composition here, and you start thinking, what can I do to that? there are some things that are well known. You can, for example, remove neutral losses, water, and that gives you a new composition. From this new composition, you can try to remove certain known subfragments. I shouldn't say subfragments at this point because we're dealing with compositions, but in reality you observe some of these fragments and they have a composition again. Yeah? But we all map it down to composition, so we can remove CH5N and we end up with this composition. That guy we can also fragment further, and we can enumerate all these possibilities. It gives rise to a pretty complex graph, and you can walk this graph in multiple ways. You can also imagine that you can first remove this fragment and then the other fragment. You can walk in different ways through this graph, but what you end up with is a complex tree that contains all the possible ways in which you can decompose this mass. And all the leaves of that are potential fragments that you might observe in your spectrum and also the inner nodes. It turns out that mapping that back to the fragments requires some thought. It turns out that you want to have some idea of which of these edges really occur. So there's lots of magic underneath, and you also want to have some idea of how likely certain things are. You can learn parts of this if you have a large enough database. And the tool that is doing all of that is Sirius 2, developed by Sebastian Böcker in Jena. And it permits the calculation of theoretical fragmentation trees from a set of spectra. It's based on a Bayesian scoring that I can't go into here because we don't have the time. But it gives you a nice overview of all the possible ways in which your composition can then be decomposed. Sirius has undergone quite a few updates by now. The Series 4 paper is about to come out, and that contains a larger data set underneath, contains more advanced machine learning, and that actually is currently the most accurate de novo identification method that you have. How does that factor into OpenMS? We've recently developed nodes for Sirius, and you can try them out probably soon, not right now. But we streamlined this whole thing so that you don't have to go through their GUI, you don't have to go through a web interface, you can just submit that. The problem with these fragmentation tree-based approaches is that they're pretty compute intensive. So spectral library search, you get a hit in a millisecond and then you're done. Sirius actually has to chew on some of these structures for a couple of minutes. So we now have a cloud set up where we can farm out these searches and scale it up to larger databases. And that is a non-trivial thing. I will finish here. I'd be happy to chat about the more intricate details of fragmentation tree-based identification with you if you like to. For now, it's probably sufficient that you have an idea of how to do it. I would like to take questions about Metabolite ID. I would just like to point out one thing before I do that, and that is a course evaluation. This course is not just sponsored by the May Institute, but also by DENBI, the German Network for Bioinformatics Infrastructure, which is why you need to do 2 evaluations. And I would really appreciate it if you could take the time for that, because that's where we get our funding from. And if you enjoyed that course and perhaps would also like to use our software in the future, then that might be a good investment of your time. Okay, but I'll stop here, and I'd be happy to take any questions.